So, hi everyone, and thank you for tuning in to this fourth installment of the worldwide seminar in addiction neurobiology. And we'll give it a couple more minutes. The number of attendees is ticking up. And we'll give it, I can turn this over to Sema Kadir, who is a PhD, a postdoctoral fellow in Melissa Herman's lab at the University of North Carolina. And we'll just wait for another 30 seconds or so for the attendees to stabilize, and I will let Sema introduce our speaker for today. And just as a reminder, if there are questions, there's a little button at the bottom of your screen to ask a question, and you can put your questions in there, and participants can vote up or down a question to be asked at the end, where Sema and I will moderate. Okay, take it away.
Thanks, Megan. So today I'm delighted to introduce our speaker, Dr. Gavan McNally. He received his PhD from the University of New South Wales in experimental psychology before going on to postdoc in molecular neuroscience with Dr. Huda Akil at the University of Michigan. He then returned to the UNSW in 2002, where he has been studying the behavioral and neural mechanisms of learning and motivation and their application in understanding addiction and anxiety. In addition to being continuously funded by the Australian Research Council and the National Health and Medical Research Council, he has received many awards from organizations such as the American Psychological Association and the Pavlovian Society. We're honored to have him here today, especially considering it's 3 a.m. where he is. And today he'll be presenting his work looking at dopamine signatures of relapse. Without further ado, Dr. McNally.
Thank you, Sema, for that generous introduction, and thank you to Megan, Marina, Eric, and Christian for inviting me to take part in this this wonderful series. It is indeed tomorrow, and I can promise you that tomorrow is indeed a better day. Before I get started and dive into the data, I just would like to highlight the people whose work I'm going to be talking about today, most notably work that's been led by, most recently, Sylvia Liu, and Philip Jean-Rita Dubussel, but prior to that, Gabriel Gibson, Joanna Yao, Zaira Millen, Ashita Prasad, and also with really significant contributions from Lucy Choi. Like many of you, we have a deep interest in the mechanisms of addiction, or mechanisms of drug taking generally, and our interest is in part practical, right? So in Australia, 4.1 million Australians consume alcohol at a daily level that places them at risk of harm. That's about a quarter of the population consuming at very risky levels. Of those individuals who meet a diagnostic criterion for an alcohol use disorder, only a few actually receive treatment, and this is despite actually an increase in the increased investment by the government in treatment centres and the availability of treatments. And of those receiving treatment, even fewer will receive a medication to assist in reducing their drinking. And this is despite the fact that there are approved efficacious medications that can and do reduce drinking and promote abstinence in the short term. But like other chronic conditions, as we all know, relapse to alcohol use and relapse to other drug use is common. It's often the modal consequence of abstinence. We're also interested in this because this is fundamentally a theoretical problem. It's a very interesting behavioral problem. Relapse is common because sustaining behavior change is very difficult. It's actually very difficult for us to change various parts of our behavior, be it changing what we eat, be it changing how much exercise we have or we engage in, and so on. And relapse is common. Behavior change is difficult because the contexts where we learn behaviors have a profound influence on acting to promote or to prevent those behaviors. Those contexts can, in addition, act as scaffolds through which other triggers or reminders of our past behaviors can come to control our behavior. And then once we've returned back to behaviors that we were trying to change, so once we've engaged in a lapse, for example, that lapse can frequently and turn very rapidly into a relapse, with a rapid return, for example, in the case of drinkers, to pre-abstinence levels of alcohol seeking and alcohol consumption. So to understand this role for context, we use, and it might help, to think about a very simple minimal experiment. When simple, I'm all in for very simple minimal things. The kind of experiment that we use is taking animals, typically rats, to make a response, typically a lever press or a nose poke, to receive some outcome. In the experiments, all the experiments I'm going to talk about today, the outcome is alcoholic beer. And what the animals learn during this self-administration training, of course, is that when they make a response, that response leads to some outcome. So they encode the response outcome association, but they also learn that when they're in that context, that response leads to the outcome. And they also learn undoubtedly associations between the context and the outcome. All of those associations are going to kind of push behavior in the same way. They're pushing behavior up. So we see the emergence of responding for and consuming the reward, the alcoholic beer. In the second phase, we extinguish that behavior, typically in a second distinctive context. It's got distinctive tactile, olfactory, visual properties as well. And we do that by removing the outcome. And the consequence of that is that behavior declines. So nose poking or lever pressing will decrease. And nose poking or lever pressing behavior will decline because the outcome has been removed from the experiment. So there is a very large contribution of what we call a generalization decrement between the training phase and the extinction phase. Simply removing the outcome depresses the behavior. But it also, responding declines because the animal learns a variety of things during this extinction. Most importantly, it learns to withhold responding, or it learns to stop responding in that context. And that learning is mediated, at least in part, by an inhibitory association between the context and the response. So that if we remove the animal from that context and we return it back to the original training context, even in the absence of the outcome, we'll see a restoration of behavior in the form, we call this restoration renewal. In this case, it would be an example of ABA renewal. In the drug self-administration literature, we call this form of restoration context-induced reinstatement, but I'm going to use those two terms, renewal and context-induced reinstatement, interchangeably. And we can also see a return in behavior if we simply allow the animal to self-administer again. That is, if we arrange that responding pays off in a retraining session, in a reacquisition session. The interesting feature about performance or behavior in that session is that the return in responding in that reacquisition session is very rapid. The animal very rapidly returns to the pre-extinction levels of responding. So a good deal of work in my laboratory, at least, has been dedicated to understanding these two influences of context. There's a protective influence of context when the animal's in the extinction context responding is being inhibited, and then there's a permissive influence of context when the animals return back to the training context during renewal, responding is promoted, and we call that restorational promotion renewal. And we've shown that these opposing permissive and preventive influences have quite distinct cellular and circuit substrates inside mostly our work, thalamostriatal and striatal pallidal circuits, but of course other people's work linking that up with cortical, thalamic and corticostriatal circuits as well. And as an example, starting inside the accumbens shell, for example, we know inside the accumbens shell that we have spiny projection neurons, predominantly dopamine 1 receptor expressing or dopamine 2 receptor expressing. We know that the dopamine 1 receptor expressing spiny projection neurons turn out to be easily differentiated on the basis of their projection target. So at least one distinct population of those neurons projects to the lateral hypothalamus, where they provide monosynaptic GABAergic input to hypothalamic GABAergic neurons. And then you have a different population of spiny projection neurons in the accumbens shell, D1 receptor expressing, also These ones project to the ventral tegmental area. But again, what they're doing down there is complex, but they're providing monosynaptic GABAergic input to VTA, GABAergic neurons. So we have these two circuits that are very similar in terms of the cells at their origin and in terms of at least some of their postsynaptic targets, different regions, lateral hypothalamus and ventral tegmental area. And it's those different circuits that seem to define the function. So, for example, if we do a simple experiment where we train an animal to self-administer alcoholic beer for 10 days, we then extinguish that behavior for 4 days. If we silence the shell inputs to the lateral hypothalamus, we can prevent the expression of extinction, or we restore the seeking behavior in the absence of the outcome. Conversely, if we were to train the animal to self-administer and then we extinguish that animal in a different context, and then we return that animal back to the original training context, and we silence the shell to VTA pathway, then we can reduce the renewal that we would see. We can reduce the context-induced reinstatement, or if you like, the relapse inside this model. So it's the circuit, not the neurochemical identity, that's dictating the shell contributions to relapse versus extinction, and renewal depends on these shell inputs into the ventral tegmental area. And of course, it's not just the ventral striatum contributing to a single form of response restoration, because if we silence that shell input to the ventral tegmental area, we can also reduce performance during reacquisition. And that reduction in relapse during reacquisition is not just because this pathway is important for self-administration, because if you silence that pathway in animals who are undergoing self-administration but have never been extinguished, there's no impact of that silencing on self-administration behavior. So dependence on descending control dissociates these two forms of relapse from self-administration. Maybe a better way of thinking about that is to say that self-administration behavior is a little bit more resilient to the removal of this shell input to the VTA. But shell to VTA seems to be very important for these two forms of relapse in this model. It's not just the shell, it's not just the nucleus accumbens. We've learned a lot about the ventral pallidum in this series of talks. I've really enjoyed all of the talks on and the pallidal data that's come up. We've been doing our own work in how ventral pallidum contributes to the forms of relapse that we study. And as you are all very well aware, the pallidum is quite diverse, but there are diverse cell types in the pallidum that, in fact, that are recruited during relapse, during renewal. And of course, they're parts of quite distinct output pathways. And we're able to at least link some of the functions of the pallidum in these forms of relapse to specific genetic identities and or projection targets. As an example, because it's going to involve the VTA, we looked at parvalbumin expressing neurons in the ventral pallidum, and we looked at FOS expression in the parvalbumin expressing neurons. We looked at PV mRNA and CFOS mRNA. In animals who have been trained to self-administer, they've then been extinguished in a second context, and then they're either tested for renewal, so they're brought into the lab and they're tested for renewal, or they're tested in the extinction context for the expression of extinction, or they were trained, they were extinguished, but they're not tested. They're just removed from their home cages on test. So this control group is, as it were, quite a nice control for the behavioral training history, the associative history, the pharmacological history of the different groups. But the important point is that what you see is animals expressing renewal or context-induced reinstatement show an increase in FOS in PV neurons. And we know that quite separately, I should say, that pallidal inputs into the ventral tegmental area are important for that renewal. We'd shown that previously. So those PV neurons and their interactions with the VTA are important for renewal, because if you take a PV Cree rat and you express the inhibitory dread unilaterally inside, in a Cree-dependent way, inside the parvalbumin neurons in the ventral pallidum, and then in the contralateral hemisphere, you express the inhibitory dread non-Cree dependent in the ventral tegmental area, You can, of course, chemogenetically disconnect VP-PV neurons, ventral pallidal PV neurons, sorry, from the ventral tegmental area with an injection of, in this case, clozapine. And we're going to compare performances of those disconnected animals to control animals. We have a variety of controls, but for the sake of simplicity, and because it's so early in the morning for me, I'm just going to talk about one control, and that control is an ipsilateral control that have the cre-dependent dread expressed in PV neurons of the ventral pallidum and in non-selectively in the ventral tegmental area in the same hemisphere, right? So one group is disconnected, one group is not, and that disconnection, chemogenetic disconnection of the ventral pallidum from the VTA is sufficient to disrupt both renewal and reacquisition. So it's no surprise then, I guess, to say that all roads lead to the ventral tegmental area, and that conclusion that that VTA and its striatopallidal inputs are going to be important for relapse is probably not novel, and we've known for a long time through the work of many groups, highlighting here, for example, the work of Jen Bossert with Yvin Shaham and Steve Marler's work using cocaine-seeking in rats or using heroin-seeking in rats, that the VTA is very important for different forms of relapse in our animal models, and that the pallidal inputs and or the striatal inputs to the VTA are important. And that aligns up with older work, with some old, beautiful older work still using fast scan cyclic voltammetry inside largely single sessions to study the impact of things like extinction and or drug-related stimuli on the release of dopamine. We also know that whatever those inputs are doing inside the VTA, it's highly likely to be complex. It's going to be largely disinhibition, but the mechanisms of disinhibition from nucleus accumbens into, for example, the VTA depend precisely on where in the VTA you are. The mechanisms for disinhibition of dopamine neurons in the VTA is different for the medial VTA versus, for example, from the lateral VTA. And this is just one of many papers from the Lamel laboratory that kind of underscoring this very important point. So we were very interested if renewal and reacquisition and presumably other forms of relapse all depend on VTA and on VTA dopamine neurons, and we're getting a handle on the precise cell type, genetic identity, and connectivity of the inputs to the VTA that are driving these different forms of relapse, what's actually happening in dopamine neurons during relapse. And not just what's happening at the level of the VTA in these different forms of relapse, what's happening in the ascending targets of those VTA dopamine neurons, what's happening in terms of dopamine release and binding in the areas that we're interested in these experiments, which are the ventral striatum. And to do that, we ran 5 different experiments. There's more than 5, but they reduced down to 5 reasonably straightforward experiments, where in some experiments we're going to take the TH-CRE rat and we're going to express the calcium sensor GCAM6 in a CRE-dependent manner inside the medial VTA or the lateral VTA, and we're going to implant fiber optic cannula above the medial VTA or above the lateral VTA, and that's going to allow us to sample calcium transients from these neurons. And then in other animals, and I point out they're different animals, right? So with one experiment's medial VTA, a different experiment is lateral VTA. And then in other experiments still, we're going to express the dopamine sensor D-light in the ventral striatum. And in some animals, we're going to express that into the medial portion, of the accumbens shell. In other animals, we're going to express that in the nucleus accumbens core. And in other animals still, we're going to express that into the lateral portions of the accumbens shell. And we're going to implant a fiber optic cannula above each of those regions to sample the dopamine transients in those different regions of the ventral striatum. And we're going to do that in separate experiments for a variety of different reasons, but mostly because we were concerned with how much behavior we're going to get out of our animals. We tend to find that the more fibers that you have implanted or the more cables that the animal's connected to, the greater the non-selective depression of the animal's behavior. That in itself, the fact that we're doing it across 5 experiments is going to create a problem, and I'll hopefully solve that problem a little bit later. So we have animals set up this way, and we're going to record, in this case, VTA calcium transients in dopamine neurons early during self-administration training. So this is either during the first or the second self-administration session. Late in self-administration training, so this is typically in the 9th or the 10th self-administration session. We're going to record them in the first extinction session. We're not going to record them thereafter in extinction, largely because the behavior goes away. We're interested in behavior. There's not a lot of behavior happening towards the end of extinction, so there's not a lot of things to link any signal to. But we're also going to record during renewal, during ABA renewal, and then the following day when the animals are being trained and being tested for reacquisition. And when you do that experiment in the medial VTA, you can express these data lots of different ways. I'm going to express the data in this case as a delta F on F, but it changes to a kernel. 0 seconds is going to be indicated here. Around a nose poke, which we call a nose poke plus. This is the nose poke that earns delivery of the reinforcer. This is 3 seconds before that nose poke. This is 3 seconds after that nose poke. This is medial VTA during early self-administration training, and you can see that we have significant inhibitory transients in the medial VTA early during self-administration training that are replaced by the end of self-administration training with significant positive transients. And those positive transients that you're seeing across that late self-administration session are pretty much lost at the end of self-administration. They don't reappear in extinction. They don't reappear significantly during context-induced reinstatement or renewal. They do reappear modestly during the test for reacquisition. So we think there's something about when you see these significant transients is primarily when the outcome, in this case, the consumable alcoholic beer, is present. It's present early in self-administration, and late in self-administration, you see significant transients. It's present during reacquisition. You see significant transients, and it's not present in these other phases, and you don't see significant transients. That profile of responding in the medial VTA is quite different to the profile of responding that you see in the lateral VTA. In the lateral VTA, what you're seeing generally are positive transients around the nose poke behavior that's earning the reinforcer, and those positive transients are persistent across all stages of the experiment. So they're present immediately in the first self-administration session. They persist into the late stages of self-administration. They're present during the first extinction session, and they're present during the 2 different forms of relapse. So immediately then you can see there's probably something different between the medial VTA and the lateral VTA in this simple task. But what that difference is a little bit hard to pin down, because it depends on exactly when in the task you decide to look. Do you look in early self-administration, late self-administration, during renewal? One of the things that struck us as interesting, though, was that we could see within subjects, we could see that renewal, ABA renewal, and reacquisition were both associated with positive transients in the lateral VTA, whereas reacquisition but not ABA renewal was associated with positive transients in the medial VTA. So we wanted to ask whether or not those differences in transients that we're seeing are actually contributing somehow to behavior. After all, we're just sticking down a single fiber and we're measuring calcium transients. There is a long way, as we all know, from a calcium transient from a lot of distance between a calcium transient and a spike. So it would be nice to know if those differences are actually relevant to behavior. And the way in which we did that was to express the inhibitory opsin halorhodopsin in the VTA dopamine neurons of TH Cree rats. We trained those animals up to self-administer, we extinguish them in a different context, and then we test them in the extinction context, and we test for extinction, we test them in the training context for renewal. For the sake of simplicity, I'll just show you the test from the renewal context. Silencing lateral VTA dopamine neurons reduces responding during renewal. Silencing medial VTA dopamine neurons does not, which is quite similar to what we'd seen conceptually, at least with the GCAMP, where we found positive transients in lateral VTA, but not significant positive transients in the medial VTA during renewal. In contrast, if silencing during reacquisition, so these are the same animals tested the next day, and the only difference between these tests is that during the reacquisition test, when the animal makes a response, it earns an outcome. Silencing either the medial VTA or the lateral VTA, in this case, is sufficient to depress responding during reacquisition, suggesting that different forms of relapse, or these two different forms of relapse, may have different dopamine mechanisms, with medial VTA contributions being linked closely to the presence of the outcome, again, and lateral VTA contributions maybe more generally. And that's a kind of okay conclusion. I quite like it, but we would be the first to concede when looking at these data that you're really trying to make inferences about behavior that's occurring across two very different baselines. That's the nature of the beast, right? Responding or performance during ABA renewal is almost always going to be lower than responding or performance during a test for reacquisition. So you're always going to have more room to move against reacquisition than you are against renewal. That is, as I said, the nature of these designs. The only reason why we might be a little bit concerned about it is that the baseline or the level of behavior that's being expressed is going to determine how sensitive or how easy it is to change. So it would be nice to figure out a way of doing that experiment where perhaps we can better match those two forms of response restoration or relapse. We can match them better on overall levels of performance to see if that dissociation between the medial VTA contribution to the two different forms of relapse actually holds up. We did, as I said, exactly the same experiments in terms of their structure, but we used expression of the dopamine sensor D-light in the ventral striatum. And these data here are showing D-light transients, again, -3 seconds to +3 seconds, centered around 0 seconds, around the nose poke that earned the delivery of the reinforcer of the outcome, what we call nose poke plus. And you can see in the medial portion of the accumbens shell that actually around that nose poke, there's like the medial VTA, there are these reductions or inhibitory dopamine transients that are present only during early self-administration training. So they're replaced by positive transients late in self-administration, and those positive transients are generally persisting across the different phases of the experiment. If you move your fiber across just a little bit from the medial portion of the shell into the accumbens core, you see something different. In the accumbens core, in this task, you see positive transients that largely persist across the different stages of the experiment. They're there immediately during early self-administration. They're present during late self-administration. They carry over into extinction. There's modest transients during renewal, and they're present during reacquisition, which is quite different, at least during early self-administration, from what you see with the medial portion of the accumbens shell. If you move your fiber over just a little bit further still in the ventral striatum to record dopamine transients in this same task from the lateral portion of the accumbens shell, you see a third different pattern. There's not a lot happening in the lateral portion of the accumbens shell early during self-administration training, but by late self-administration training, we're seeing strong, significant positive dopamine transients that are lost during extinction, and they really only come up again during reacquisition. So across the ventral striatum, we find broadly 3 different profiles of dopamine transients that vary depending on when in the particular phase of the experiment you decide to look at your recording, right? So early, late, extinction, so on and so forth. So if you put all of that together if you put together your medial VTA calcium transients and your lateral VTA calcium transients and your medial shell, core, and lateral shell dopamine transients, and you don't care for a moment that they're coming from different animals, and you don't care for a moment that they're reporting their different senses, if you just put them all together and ask, are they going in the same direction or not across these different common stages of the experiment, the answer is, well, it depends. Sometimes, for example, during late self-administration and certainly during reacquisition, you have what looks to be a homogeneous or unified system. Everything's going in the same direction, from medial VTA up to lateral portion dopamine transients, from calcium transients in medial VTA up to dopamine transients in the lateral portions of the accumbens shell. Generally, it seems that these different compartments of the mesolimbic dopamine system are moving in the same direction. That's also true in reacquisition. But at other times, you see what we've come to expect from the more modern research on the mesolimbic dopamine system is you see a very diverse and heterogeneous system, especially early during self-administration training, where we have different compartments or different elements of mesolimbic dopamine compartments showing quite different changes. But that's just eyeballing it, right? That's just us aligning these things up and me drawing shapes in PowerPoint to exploit grouping effects so that that's what you see. But surely there's got to be a better way that we can assess these data. The problem, there are two problems that we could think of. You can probably think of many more. We could only ever come up with really two big problems. The problem when trying to assess these data any further is firstly the multiple comparison problem. If we wanted to compare within a region, this is the medial shell dopamine transients. If we wanted to compare within the medial shell across the 5 different stages of the experiment to ask which stages are similar with each other and which stages are different, for the medial shell alone, we'd have to make 10 pairwise comparisons. And that is, of course, assuming that we're not interested in differences in the shape of the waveform. So that is assuming we reduce down the quite interesting waveforms to a single summary statistic, be it area under the curve or peak delta F on F. That's okay though, right? We could do that, and we have statistical techniques, of course, that allow us to control for the fact that we'd be making 10 multiple comparisons here, 10 multiple comparisons for each region, so 50 comparisons. But the bigger problem that we confronted when trying to think about these data was, how is it even that we can compare between regions, between senses, potentially even between different animals? Assuming that you could, you'd have 5 brain regions, 5 stages, and 2 senses. You'd have about 300 different pairwise comparisons, assuming that one could validly compare our senses, which are biophysically, of course, very distinct, and potentially more importantly, they're just reporting fundamentally different biological processes. They're reporting biological processes that we hope are related to each other. We kind of hope that when we record a calcium transient, even crudely with a fiber in the rat, in the VTA, we kind of hope that a calcium transient in a dopamine neuron in the VTA is going to somehow be related to a D-like transient in the ventral striatum. But there's careers worth of effort between those two measurements, right? So you can't simply compare between the senses because they're different. It's even problematic comparing between the different regions because they're different. So we have to somehow solve this problem of correspondency. Fortunately, we didn't need to solve it. That our colleagues in cognitive neurosciences solved this problem some time ago. And if you're not familiar with representational similarity analysis and you're a behavioral neuroscientist, I highly recommend it. It's quite fascinating. The key original paper, of course, is the neuron paper by Krieger-Korda, but then he's gone on to provide really accessible reviews and toolboxes. And we're going to basically take off-the-shelf representational similarity analysis techniques which are developed for analyzing fMRI data, and we're just going to human fMRI data mostly, and we're going to apply them to our photometry data set. And the idea here is that what we're going to do is to calculate for each animal, we're going to calculate the correlations between the different pairs of waveforms of our sensors. So this could be, here's 3 waveforms. I'm not sure which ones from which animals or which region they actually are. We calculate the correlations for each animal between the different pairs of waveforms, and then instead of reporting or assembling a correlation matrix, we compute, which reports similarity, we compute a dissimilarity matrix. So we assemble 1 minus the correlation coefficient. So we deal with correlation distances. And this shows you what such a matrix might look like. This is probably medial VTA, I think, or it could be lateral VTA. It doesn't actually matter because it's just an example, where each row is an animal and the colors indicate the correlation distance, 1 minus the correlation distance, where blue means 0 distance or highly identical, red means a distance of 2, therefore perfectly dissimilar. We can assemble those dissimilarity matrices for the brain regions. So for example, for the medial VTA and for lateral VTA, and we can report those matrices like this. And when you look at these matrices, you're seeing a lot of blue, and generally a lot of blue is telling you that across the different phases of the experiment, for example, in the medial VTA, the GCAMP signals are reasonably similar. They're highly similar between late acquisition and reacquisition. They're not so similar between late acquisition and early acquisition. And so you can look at these to try and get a sense of the relationships between the different phases. But it's easier to reduce that dimensionality down to 2 dimensions using multi-dimensional scaling, where you can then more clearly see the relationships between within medial VTA, between the GCAMP signals across the different phases of the experiment. And what you can see is that early acquisition is different to late acquisition, which is certainly different to extinction and renewal or ABA renewal, context-induced reinstatement, and that late acquisition is reasonably similar to reacquisition. The lateral VTA is a lot more blue across the different phases of the experiment, and the MDS kind of supports that increase in similarity. The different phases of the experiment are more closely related to each other. Early acquisition is very closely related to late acquisition, which is closely related to reacquisition. Renewal is kind of in the middle, and extinction is out to the side. But overall, there's high similarity within the lateral VTA across the different phases of the experiment. And that ought to be true because, as I showed you, I'll go all the way back, sorry. In the lateral VTA, you're getting positive transients across all different stages of the experiment, and that's what that matrix is picking up on. That's not kind of the interesting part of these analyses, though. The first order representational dissimilarity matrices are kind of interesting, but the reason they become more interesting to us is that we can go 1 step further. We can compute these first order matrices for the medial VTA and lateral VTA, as I just showed you. You can compute them for the D-light transients in the lateral shell in the accumbens core and the medial portion of the accumbens shell. But then we can compute higher order or second order matrices, where what we're comparing now is not the underlying activity waveforms. We're not trying to compare a D-light sensor in the medial portion of the shell to a GCAMP waveform in the medial VTA, but rather what we're going to compare is the first order matrices. We calculate the correlation distances across these first order matrices. And these second order matrices, because they're directly comparing the first order ones and they're not directly comparing the underlying activity waveforms, allow us, we think, to make, you might disagree, we think they allow us to make fair comparisons, valid, certainly fair comparisons across different sensors, across different brain regions, across different behavioral stages. They're helping us, as it were, to understand the distinctions. What distinctions between these different phases of the experiment does the mesolimbic system, dopamine system, care about? and what distinctions does it not care about. When you calculate or when you compute those second order matrices, you end up, you can do them any number of ways. We calculated one for our different brain regions, and what we find is that within the Mesolimbic dopamine system, the second order matrix is shown up here. There's quite a bit of blue and there's some yellow. And when you look at the multidimensional scaling, it falls out. That is to say that there are at least 3 distinct Mesolimbic dopamine circuits being linked to behavior in this task. And this is regardless of the particular phase of the experiment, right? So this is ignoring phase of the experiment. We're just looking at regions. The medial VTA is very similar to the medial portion of the accumbens shell. The lateral VTA is unsurprisingly very similar to the accumbens core, and the lateral portion of the accumbens shell is quite different to the rest of these compartments. And that's also clear up in the matrix, where you can see that for the accumbens shell lateral, there are high levels of dissimilarity across these different experiments than there are for any other compartment or element of the Mesolimbic dopamine system. So a lot of the variance, there's a lot of dissimilarity between the different components of the ventral striatum. We've got medial shell, core, and lateral shell, but really the unusual one here is the lateral shell. And that tells us, what this matrix is telling us is that the medial shell and the medial VTA are sensitive to common features of the task across the entire experiments. The core and the lateral VTA share a sensitivity to a different but common to them set of task features. And the accumbens shell lateral is sensitive to unique task features. Okay, you can do exactly the same thing when you're looking at behavior. So now, instead of computing the second order matrix across brain regions, we computed across the different experimental phases. There's a lot of different colors in here, so we'll reduce it down using multidimensional scaling. And one of the things that's interesting is when you do the multidimensional scaling is that the 5 different phases, the 5 different behavioral phases of the experiment are located in different spaces, right? They're different to each other as far as the Mesolimbic dopamine system is concerned. It's animals being put into boxes and being tested in the same apparatus, where we're changing the associative history of the context, where we're changing the reinforcement contingencies, and of course the Mesolimbic dopamine system is clearly sensitive to that. It's picking up on that. So we have self-administration as being distinct to late self-administration. We have late self-administration as being distinct to extinction. Extinction is an interesting point in these data, because the coherence in the signals that we see building up across the course of self-administration are rapidly lost during extinction. Extinction is, as it were, a kind of fracture point in these data. Renewal, our one form of relapse, our context-induced reinstatement, although sensitive to a dopamine receptor antagonist, to silencing dopamine neurons, like reacquisition, sensitive to dopamine receptor antagonist and the silencing dopamine neurons, is represented quite distinctly within the Mesolimbic dopamine system. The signature for renewal, the activity signature for renewal is quite different to the activity signature for reacquisition. And that's well supported by our inhibition data, which suggests, for example, that silencing the medial VTA doesn't really have much of an effect, or is able to dissociate, I should say, between these 2 different forms of relapse. And it suggesting that we think this is interesting, because we think that the existence of these 2 different signatures suggests that these different forms of relapse are being mediated by quite different changes across the system. And now we're quite interested, for example, in how extant medications against relapse are shifting. How do they affect either the difference between the signatures of these 2 different forms of relapse? So how do they affect the underlying activity signatures across the Mesolimbic dopamine system itself? The other thing that stands out from this behavioral matrix, of course, is that reacquisition is reasonably similar to late self-administration. Certainly when you look inside the matrix at the highest levels of similarity across the different phases of the experiment are between reacquisition and late acquisition. This is what someone from a learning background would simply call savings, but it's a demonstration of savings in mesolimbic dopamine activity signatures. Late self-administration is reasonably similar to reacquisition. Certainly it's more similar. Reacquisition is more similar to late self-administration than it is to early self-administration. So whatever, as it were, our decremental treatment is doing, whatever extinction is doing to depress behavior, and whatever removal of the outcome during extinction is doing to depress behavior, it's not conferring any long-lasting protection on the animal, and that the mesolimbic dopamine system, much like the whole animal and the animal's behavior, rapidly returns in this single reacquisition session to how it looked at the end of self-administration training rather than how it looked at the beginning of self-administration training. So that's about all that I want to say. It's probably all I have the energy to say. Dopamine is important for causing relapse, and that's quite obvious. This role is linked to multiple descending inhibitory inputs from the striatal pallidal system. I showed you examples from the accumbens shell and from the ventral pallidum, but there are also inputs from hypothalamus and others still. And even under the quite impoverished, very limited and minimal conditions that we're studying these circuits, there are important nuances in the role of dopamine and mesolimbic dopamine in relapse that depend on the specific circuit, that depend on the presence versus absence of the reinforcer, and that depend on the animal's behavioral history. So whether these nuances that I've talked about are specific to our behavioral preparation, I hope they're not, but whether they are or whether or not they reflect a more general functional properties of the mesolimbic dopamine system remains to be determined. So you might be interested in what happens with other reinforcers. I don't know. You might be interested in what happens with extended access. I don't know, although I would like to know. You might be interested in what's happening in the dorsal striatum. We can't say just right now. More generally, though, the modern toolbox is providing us with very powerful approaches for mechanistic deconstruction of circuits, of course, but behavior, as we all know, is very rarely a product of a single circuit. And even if it were a product of a handful of circuits, there are potentially as it were, many modes of activity within those circuits, within the mesolimbic dopamine system. Whether or not you view it as a unitary system or a diverse heterogeneous system depends on where, in these experiments, where during the experiment you're looking. At sometimes during the experiment, late self-administration and reacquisition, everything's pushing in the same direction. At other times, they're going in each compartment, or different compartments are going, as it were, in their own direction. And I would just say that better understanding of normal behavior, because ultimately normal behavior is also very interesting to us, and better treatments of pathological behavior are going to require approaches that better bridge cellular circuit and behavioral levels without sacrificing any of the power that we can get from each of those levels. And that is all I have to say. I'd like to acknowledge again the wonderful work of Sylvia Liu, Philip Jean-Richard de Bruxelles, Joanna Yao, Gabriel Gibson, Ashita Prasad, Lucy Choi, and our collaborators, Colin Clifford, who taught us about representational similarity analysis, my colleague Simon and colleagues at UNSW, Melbourne, Andy Lawrence and Erin Campbell, Newcastle, Chris Dias, and Amsterdam, Nathan Marchand, all of whom have been very helpful in helping us get some of this work done, or certainly setting us right on our thinking about it, and any errors in that thinking and any errors in that presentation are owned by me and not owned by them. So thank you very much.
Wonderful. Thanks so much. And so we have a couple of questions in the chat. I might, give people a second just to type them if there are any more. I really quickly wanted to ask one to get started, and that was when you're defining the early versus late training, how many sessions are going into each of these measurements, and are they the same number of trials that are going into the sessions that you're using to calculate the transients for the early versus late? And is the behavior changing more across the early training phase, is there more variability in the transients compared to the other phases where you're comparing?
There's certain in terms of the individual shape of the transients, there's not so much variability in those in their shape if it's early versus late. Behavior picks up pretty quickly in this task, so by about the second session they're responding at quite high levels, so that we don't see the same kind of gradual increase in responding in this task that you might see in others. So there may well be more as it were responses during the late self-administration phase than the early self-administration phase, but it's not a very, very large difference. It's certainly not as large as you might find in other preparations. That is a problem, of course, when you're trying to compare the two forms of relapse between renewal and reacquisition, and that is just inevitable. There's always more behaviour during reacquisition, but there's always more behaviour when the reinforcer's around than when the reinforcer's absent, which is actually kind of hard to control. It's one of the reasons why we're reluctant to make direct comparisons between different phases, but we don't mind making comparisons across the second or using second order RSA to make those comparisons, right? Where maybe we can get around some of those problems, but yeah.
Okay. And Sema, I'll let you moderate the questions from the audience because there are a couple of really good ones in there.
Yeah, so to start off, first of all, great talk. But Kurt said, great talk, Gavin. I'm curious about the apparent lack of activity in both the VTA and dopamine release throughout the ventral striatum during context renewal of instrumental responding. This seems to counter intuitions that dopamine activity and release are in turn critical for reinstatement and relapse. I was wondering if you could elaborate on why in this setup there's minimal activity and how to reconcile that with the apparent halo-redox syndrome.
Yeah, so there's not a lot. I don't know, Kurt. That's a good question. In terms of magnitudes of these signals, there's not a lot. There is within subjects, for example, in the VTA, there are within subjects significant positive transients in the VTA with calcium transients during renewal in the lateral VTA, but not the medial VTA. Certainly with the D-light signal, there's probably more robust evidence for dopamine binding across the medial shelf, certainly during renewal, and to some extent the core also. They're just not very large. So I think overall you can reconcile that reasonably easily with the silencing data, because the silencing data shows something similar. The behavior is certainly, as you well know, if you administer a D1 or a D1 receptor antagonist, you'll knock the behavior out completely. So the behavior is very, very sensitive to dopamine, but we are not seeing big juicy signals, I agree. And I think certainly during renewal, and I think one of those reasons is the reinforcer is just not there, right? The presence of the reinforcer does a whole bunch of wonderful things to behavior, which I'm going to assume that means it also does a whole bunch of wonderful things to circuits as well. I'm not sure if that answers your question or not, Kurt, but that's a very good observation and a very good question.
Great, thank you. John says, thanks for a fascinating talk. Did you also look at dopamine transients evoked by the drug itself? If there was evidence for altered drug function, this could then trigger mechanism, ultimately providing an explanation for your
So as in prior to the experiment, I would imagine, is the question there. Would that be a fair interpretation? Yeah.
I think so.
I believe it's a question from Christian as well.
Yeah.
Oh yeah.
No, we didn't. That's a really good idea. I wish we had. So we could have just taken those animals and done them at rest, where you're probably hard to know what you're seeing, but you could have given them different events to see different rewards or different events to get a sense of how it might be, how it is they may be changing. We haven't done that. That's a very good idea, though. Thank you.
Next, Alexa says, great talk. Are there any differences between early and late?
Good question, Alexis. I wish I knew. That's something we care deeply about, for a variety of different reasons. Behaviorally, the learning that's going to be occurring during extinction will be much greater during early extinction than during late extinction, just as presumably the learning that happens during self-administration is greatest early during self-administration and late self-administration. That may well be why this extinction and early self-administration are so different. The problem is that when you kind of look in the fourth session, the third or fourth session, and you kind of turn on your computer and you start making the recordings, you just get nothing. There's just, you know, tumbleweeds. There's not a lot of behavior by that stage. Certainly the animal's not doing very much. You put it in the box, the animal circles around it, grooms for a little bit, and then by the fourth extinction session will be resting, and there's not a lot of dopamine transients. So it's kind of hard for us to kind of get at that properly. Again, what you really want to do is get the behavior to about the same level. Potentially you could get around that by introducing 2 responses. As an example, something we're interested in doing, you reinforce the animal on 2 responses, and then during extinction, you continue reinforcing 1 response. You don't reinforce the second response, and then you see what happens to that second response across those different extinction sessions. And you'll have a nice overall baseline of behavior to make those comparisons, because you'll always have that other response in there that's not been extinguished. So we don't know. It's an excellent question, and it's something that we're interested in, but you probably need a couple of responses and ideally 2 different outcomes to get at that, and that's something that we're trying to do.
That's fascinating. So next, Ankush wanted to know, how late after extinction do you check for renewal? And do you think the amount of time after extinction has an effect on
the good question. We test the next day. We're always in a bit of a rush here. Not so much. We're not so much in a rush. It's just that's just the way that we do it. We have tested, I think, up to about 2 weeks later. We personally have never seen much of an influence of the passage of time. We've never systematically examine that. Like we haven't gone out to 49, 50 days, 60 days, 70 days like the brave heroic souls do in those incubation experiments. Theoretically, of course, you would expect that with the passage of time after extinction that behavior will recover anyway due to spontaneous recovery. You might then expect that will summate with any restoration due to renewal. So maybe you'll see an increase in more renewal 4 weeks after extinction than 1 day after extinction. Spontaneous recovery is reasonably elusive under the conditions that we're studying here for precisely the same reason I talked about with outcomes before. So I don't really know, but again, I think that'd be quite interesting. If you could do that experiment properly, the experiments that I've talked about, the designs today, aren't really going to let you do that. You need to be a little bit more clever about them.
Makes sense. And then I think lastly, Kyle says, amazing job. I'm very interested in the modeling you did. Could you determine if there are subgroups in the behavior, such as high and low risk self-administration, and then run the same models and look at differences between subgroups?
Sorry, Sam, I missed the beginning of that question. Could you read it again?
Yeah, I'm sorry. So Kyle's asking if you could determine if there are subgroups in the behavior, such as like high and low risk self-administration, and then look at the models for region.
Yeah, great question, Kyle. We haven't done that. We perhaps ought to have. We don't see as much variability in the behavior, I think, that we would probably need to do that. That doesn't mean we couldn't see it, right? We just change the schedule. We can introduce a lot more variability, I think. And I think we'd need to do that. But yeah, you could if you had that variability from the outset, you could then see exactly how those signatures are potentially changing across the experiment as well. We unfortunately can't do that because our animals are too similar. There's not spreading out enough. We don't have low responders or high responders. Generally, everyone's a high responder, if you like. But that's a really good idea.
Kind of going along with that, I wanted to ask, have you looked at sex differences? Because that would definitely introduce variability if you're testing females, just because females are...
I'm not sure that they are. I'll stand up for that one. I'll kind of maybe push back a little bit on that. I'm not actually sure that they are all that more variable. Certainly in our hands, they're not. They kind of depends on how you do it. They kind of go along. In our hands, they don't look that different to males most of the time, and certainly not more variable in the experiments that we do. These were all male rats, but I can tell you that we've done all the behavioural experiments and the antagonist experiments recently in female rats. They look exactly the same. Whether or not you could pick out sex differences using the RSA. I think it'd be really interesting, actually, because that approach to the analysis is it's pulling out different things to simply carving individuals up into groups, right? So yeah, I think that'd be really cool to look at.
Yeah, thank you.
Great, and I think that brings us to noon. But I want to thank everybody for the really engaging discussion, the wonderful questions, and definitely for Gavan for waking up at 3 in the morning and giving a great talk in spite of the hour. And I think next week we meet back here, and it's Serge Ahmed. All right. Thanks so much.